Hello everyone, welcome back to Infinity Learn NEET channel. My name is Kiran and I am your Botany faculty at Infinity Learn. Students, in this class we will be discussing about all the important PYQs from Principle of Inheritance and Variation. This is your chapter number 5 from class 12. So let's check out what are the important questions NTA has already asked in the previous history of NEET exam, and I will really appreciate if you will participate in the session as well. So the question goes, but before that, if you are new to our channel, do consider subscribing to Infinity Learn NEET channel, my dear students, for more such videos and content in future. That is definitely going to help you in crack your NEET exam. Next, also do consider visiting to our website, my dear students, infinitylearn.com website, and check out the amazing courses and the services, the products that we offer. You can also register yourself for an IELTS master class for your NEET exam, for your NEET preparation, my dear students. What are the features that we offer at our website? You will get free master classes. You can also attempt one of the Infinity Learn test series, one of the tests as free. Under Infinity Learn test series, you can also take 2 mock tests free, and there are 14 free previous year question papers along with the correct answer key and the solution. This is already we are offering for free, so do consider visiting to infinitylearn.com site and check out the amazing things that we are offering over there. Next, okay, so thank you very much students for cooperating with me, and we are back now with the very first question. How many true breeding pea line varieties did Mendel selected? Just give me a moment again. How many true breeding pea varieties did Mendel selected as pairs which were similar except in one characters with the contrasting trait? This question was asked in NEET 2020. Come on, give it a try. Hello Savita, very warm welcome to the class, my lovely kid. So what do you think can be the probable right answer to this question? Any guesses? Yeah, that's absolutely true. 14 true breeding pea varieties. Excellent. For the 7 characters. Next, this question was asked in NEET 2017. Among the following characters, which one was not considered by Mendel in his experiments on pea? Any guesses? Savita, my lovely kid, any guesses? Any guesses, my dear? Okay, then let me help you out over here. Trichome, glandular or non-glandular was not at all any character that was chosen by Mendel in his experiment in a pea plant. Seed color, yellow or green, pod shape, inflated, constricted, stem, tall or dwarf. These were the character chosen, but the first one is not the one. Next, this question was asked in fact in NEET 2017. Which one from these given below is the period for Mendel's hybridization experiment? I want everybody to give importance to this. My dear students, we generally tend to skip out the data, the material, the text that is written before inheritance of one gene, right? It's related to Mendel and his work, but let me tell you that topic, that whole paragraph, that whole page is extremely important. In the previous history of NEET exam, several times NTA has asked question from that line, right? And I have already shown you some of the question. One was on the number of true breeding line, number of characters chosen, what were the characters, right? Where were those characters, and even the time period between which Mendel did his experiment. So this is an important paragraph. So I hope with these important PYQs you will be getting that, not to skip that paragraph, that page, the text that is before your inheritance of one gene. So the answer is 1856 to 1863. Please remember this. Please remember this. So NTA has already asked this and might ask again. Who knows? Maybe in NEET 2022. Right? Hello uncle, very warm welcome. Next question. This was asked in 2015. In his classic experiments on pea plant, Mendel did not use. In his classic experiments on pea plant, Mendel did not use. So can anybody guess what Mendel did not use? Which of the following characters Mendel did not use? Can anybody guess over that? Which of the following are the characters that Mendel did not choose? Any guesses on this, my kids? Which of the following characters that Mendel did not choose? Excellent. Pod length was never the character chosen by Mendel for his experiment. Excellent, Ankul and Savita. Next was asked in 2015. How many pairs of contrasting characters in pea plant were studied by Mendel in his experiment? So please notice till here the last 5 questions I have been discussing are all on that introductory paragraph, that very first topic. Good afternoon. So can anybody tell me how many pairs of contrasting characters were chosen? Anyone? Yes, excellent. It is 7. Next, this question was asked in NEET 2020. Identify the wrong statement with reference to the gene I that controls the AB blood groups. The gene I has actually, my lovely kids, 3 alleles. That is absolutely correct. So you are supposed to identify the wrong statement. This is correct. And what are the alleles? IA, IB and small I. A person who will have only 2 of the 3 alleles, yes, because we are diploid organisms, so we can have only 2 alleles in a pair of homologous chromosome on the same locus. When IA and IB are present together, they express the same type of sugar? No. Individually, both IA and IB will express itself, and the sugar is going to be AB, not the same type. So IA will express itself and will show the respective sugar, and IB will also show the its respective sugar. So the blood group is going to be AB and not just either A or B. That's an example of codominance. Right, my lovely kids, is this much clear? Okay, now let's talk about the next statement. Allele I does not produce any sugar. Allele I does not produce any sugar is in fact correct, and that is why in the case small I, the blood group is O, no sugar. So both the sugars A and B will be present here, and the blood group is going to be AB. So which of the following is wrong? C is wrong. Next question. This was asked in NEET 2019. Excellent. This was asked in NEET 2019. In Snapdragon Antirrhinum, the red flower are crossed with white flower, and in F1 generation, all pink flowers were obtained. So this is an example of incomplete dominance. You remember, my dear students, deviation for Mendelism. This has been discussed. Next, so when pink flowers were self, the F2 generation showed white, red and pink flower. Chose the incorrect statement from the following. So law of segregation does not apply in this experiment. In fact, law of segregation is one of the most universal law in the entire biology, my dear students. There is no exception to it. It is always followed, right? Why this pink color is coming, why all the progeny is pink in F1 generation? That's not because of segregation not getting followed. That's because law of dominance is not followed completely. So because of incomplete dominance, the gene that is responsible to provide red color is incompletely dominant over the gene because of which, my dear students, white color flowers are coming, and because of which you're getting pink flowers, right? Excellent. So this is in fact correct. So choose the incorrect statement. So this one is incorrect because law of segregation is definitely applied. This experiment does not follow principle of dominance, yes, because it is actually a case of incomplete dominance. Pink flower in F1 is due to incomplete dominance, and the ratio of F2 is 1:2:1. 1 out of 4 will be red, 2 out of 4 will be pink, 1 out of the 4 will be white. Absolutely correct, absolutely right. Only first one is incorrect because it says law of segregation does not apply. That's wrong. Law of segregation is applied. This question was asked in NEET 2017. The genotype of a husband and a wife are capital IA, capital IB, and
so of husband it is this, and of wife it is capital IA and small I. Among the blood group types of their children, how many different genotypes and phenotypes are possible? Now let's check out that, my lovely kids. So let's start over here. Let's do some homework here. IA, IB, IA and small I. So how many types of gametes are possible? So two types of sperms can be produced. One can carry capital IA, another can carry capital IB. Here also two types of eggs can be produced or ovum, one carrying capital IA and another carrying the small I. Now let's talk about the combination. So whenever this sperm fuses with this one, what will happen? It will be capital IA, capital IA. Whenever, okay, take this. Whenever IA fuses with small I, what will happen? It will be IA, small I. And what are the other possibilities, my kids? When IB fuses with this one, what are the possibilities? IB, okay, IA rather I can write instead of this, because by default you write A first. By default you write A first. This is going to be IA and IB. Right, kids? Next color I should choose is orange for IB and I. This is going to be IB and I. So what did you notice? What did you notice, my kid? You will notice how many genotypes are possible. How many genotypes are possible? This is genotype number 1. This is genotype number 2. This is genotype number 3 and this is genotype number 4. All are different from each other. How many genotypes are possible? 4 genotypes are possible. 4 genotypes possible. Now let's check out, my lovely kids, how many types of phenotypes possible. Capital IA will give what? That's absolutely correct, Savita. Thank you. That's absolutely correct, Uncle. Thank you. Now I'm just explaining it. Let me know if you're happy with the explanation. So this is going to produce an A blood group. This is also going to produce A blood group. This is going to produce AB blood group. And this is going to produce B blood group. So there are 2 A's, 1 B and another AB. So total 3 types, because both these equals to A only. So 4 types of genotypes and 3 types of phenotypes are possible. Right, kids? Okay, so the answer is 4 genotype and 3 phenotypes. Excellent. Good. I really appreciate that you were able to give the answer before I could explain. That's absolutely amazing. Keep this good work. Okay, keep doing it. NEET 2016. A tall true breeding garden pea plant is crossed with a dwarf one. So it is like capital T crossed with small t. When F1 were self, the resulting genotypes were in the ratio. So this question is simply based on the result of, or phenotypic result, phenotypic, genotypic, they are actually talking about together, result of Mendel's monohybrid cross. So you already know in F2 generation, the genotypic ratio is what, my dear students? The genotypic ratio is actually 1:2, 2:1, right? And in that, if you notice, how many of them are tall? So there are actually 3:2. If you remember the F2 generation, my dear students, in Mendel's monohybrid cross, the genotypic ratio is 1:2, 2:1, but the phenotypic ratio is 3:1. That means 3 out of 4 are tall and only 1 out of the 4 is dwarf. But the 3 which are out of the 4 tall, there is also bifurcation. Only 1 out of the 4 is actually pure tall and 2 out of the 4 are heterozygous tall, and that's what is written over here. So 1 out of the 4 is pure tall homozygous, capital T. 2 out of the 4 is heterozygous tall, capital T, small t, and 1 out of the 4 is dwarf, small t. So that's nothing simpler than this you can expect from genetics. Excellent. Next question, guys. This was asked in 2015. A gene showing codominance has. A gene showing codominance has. So what do you mean by codominance? So let me help you in understanding the difference between codominance and incomplete dominance, my kids. Please listen this carefully. In case of incomplete dominance, my dear students, none of the gene is completely dominant over the another one. So it is actually incorrect to say that this gene is dominant and this one is recessive. So the concept of dominance and recessive actually fails in case of incomplete dominance that you have already seen in antirrhinum majus or snapdragon plant. But when we talk about human blood groups, which shows co-dominance, not in all types of genotypes, but the specific genotype. If you talk about the specific genotype, capital IA and IB, both these alleles, my dear students, both these genes, both these alleles, capital IA and capital IB, they are equally dominant over each other. So here also you can't say this one is dominant or this one is recessive. Both are equally dominant and both will express themselves in the progeny. Right? Yeah. So the blood group won't be A, won't be B, it will be AB because both the type of sugars will be present over here. So this is an example of codominance. So in codominance, in heterozygous condition, both the alleles, both the genes express themselves, and in incomplete dominance, none of them express themselves. So that means in case of incomplete dominance in F1 progeny, my dear students, F1 progeny does not, any of the hybrid of F1 does not represent to any of either of the parents. We can see only in population. Yeah, it's a case of multiple allelism also, but right now we are not talking about that, we are talking about codominance. Okay, so that's absolutely correct that human blood group is an example. That this is also one of the questions that I'm going to give you a practice. That's a good one, that human blood groups can show you an example of dominance as well as codominance as well as of multiple allelism. I'm going to talk on that at a length. Just give me some time, uncle. So a gene showing codominance has both alleles independently expressed themselves in the heterozygous condition. Beautifully written line. Both alleles independently express themselves in the heterozygous condition, and this will become opposite when it is the case of incomplete dominance, that both the alleles won't be able to express themselves in the heterozygous condition. Clear, guys? Next question. This was asked in 2015. Alleles. How do you define allele? So very simply, it is given in your NCERT as well, my dear students. Alleles can be easily expressed as they are the different molecular forms of gene, or they are the alternate forms of gene. For example, capital T, small t, both are alleles. For example, capital IA, capital IB and small I, what are these? These are alleles, alternate forms of the genes. Next question. This was asked in 2015. A man with blood group A marries with a woman with blood group B. What are the possible types of phenotype? Now, to be honest, my dear students, here you need to do a homework, right? Although if you know the answer, definitely the absolute the obvious answer is A, B and O. But this, if you already know this fact, but if you do not know this fact, then let me help you out in how we can calculate. So actually the information given in the question is incomplete. They did not explain what type of, they just gave the phenotype. They did not mention the genotype of the parents. So we have to assume. So this blood group A can be because of either capital A, capital IA, or it can be because of capital I and small I. And this blood group B can be because of capital IB and IB small I. Is this clear, my lovely kids? Yep. Okay. So let me do this now. Okay. So let's take the first case, male and female. So the first case is, let's take this case, IA, and I am taking female to be this one, IB, right? So what will happen in this case? Okay, let me draw all the cases. So in this case, the female, I should maintain the order rather. I will write male first and female last. So here also male and female. The male, I'm keeping the male same, IA, but now I'm changing the genotype of the female. It can be IB and small I. Similarly, now I'm taking the another possibility of male, IA, small I, and again IB, and in this case IA, small I and IB, small I. Okay, so let's see the possibilities. What are the possibilities, my dear students? So capital IA, this male I'm talking about, this capital IA has two possibilities, right? Both. So see here, both the genes or both the alleles in male is same. So I'm going to explain this question with the help of fork line method, which is very simple and very convenient, right? So I'm going to draw only one fork line for the male because both the genes are same. And where is the variation? Even the variation is not there in the female as of now. Both male and female are going to produce, my dear students, only same type of gametes. Both male and female are going to produce same type of gametes. So only IA will come from male and only IB will come from the female. So all the progeny will be IA, IB. That means the blood group is going to be AB. The blood group is going to be AB. Next question. Here if I look about, so there is only one type of a gamete possible, sperm possible in male, and two types of egg possible in female. So I will draw only one fork for the male, and only one fork is enough because only one type of sperm will be produced. But this sperm can fuse with two types of eggs. Okay, so it can be capital IB and small I. So as a result, in this case it will be IA, IB, and in this case, my lovely kids, it will be IA and small I. So what are the possibilities? Blood group will be AB again, one possibility, and it will be A in the next possibility. Now let's check out over here. Here the female is having or going to produce only one type of an egg because both are IB, IB. So let's start with female IB, and there are two possibilities of the sperms that can fuse with it, capital IA and small ia. So as a result, it will be IA, IB, and in this case it is going to become IB, small I. So what is the blood group possible? AB, and another blood group possible is B. Now let's see the next case. Here both the male and female can produce two types of gametes. So let's start from male, and we are going to produce two fork line this time, capital IA. So I'm teaching you at the same time how to solve such question with the help of fork line. Okay, so capital IA, two forks, it can either fuse or fertilize with an egg with capital IB or with small I, and the small I can of the sperm that is containing small I from male can fuse with an egg of capital IB or small I. So in this case it will be IA, IB. In this case it is going to be IA, small I. In this case it is going to be IB, I, and here it will be small I, small I. So what are the possibilities? AB. What are the possibilities, my dear students? A. What is the possibility? B. And what is the last possibility? O blood group. So if you notice, all the four types of blood groups you can easily see in the last case. So because it is not mentioned what type of genotype these parents have, so we have to consider all the cases, and the answer is A, B, AB and O, which can easily be explained with the last cross. Is this clear, my lovely kids? I have shown you all the possibilities and also taught you how to use the fork line method. Clear, guys? Any doubts in this? Uncle, Savita, if you do not have any doubt, give me a thumbs up. Next question. This was asked in NEET 2013. Which idea is depicted by a cross in which F1 generation resemble both the parents? So multiple time I have repeated in the class, whenever F1 generation, listen this very carefully, thank you. Whenever F1 generation, my lovely kid, whenever F1 generation represent either of the parents, it is the case of dominance or complete dominance. When F1 generation does not represent to either of the parents, it's a case of incomplete dominance. And when F1 generation resemble both the parents, it's a case of codominance. Now please remember this conclusion, okay? Codominance, because the F1 generation resemble both the parents. Next question. This was asked in the latest NEET 2020. Experimental verification of the chromosomal theory of inheritance was done by. So chromosomal theory of inheritance, my dear students, which actually show a parallel behavior between genes or the so-called factors by Mendel and the chromosome behavior, is so parallel during meiosis or cell division. right? It was given or proposed by Sutton and Boveri, but the experimental proof was given by Morgan. Correct? Excellent, guys. Next is NEET 2019. What map unit, or in centimorgan it is the unit, is adopted in the construction of genetic map? So remember, my dear students, while constructing a genetic map, my dear students, while keeping the position of the genes and defining the relative position of one gene with the another, such kind of genetic maps, right, can be obtained, and the idea was actually given by my dear students who actually worked on it in a detailed way is the student of Morgan, that is Alfred Stewart one, and again one of the questions is going to come, don't worry about that. Thank you, everybody. So what's the question is? The question is how do you define them? So the definition says a unit of distance between two genes on a chromosome is represented by 1% crossover. A unit of distance between two genes on a chromosome is represented by 1% crossover. Next, this was asked in NEET 2019. The frequency of recombination between gene present on the same chromosome as a measure of the distance between gene was explained by. So let me tell you, my dear students, it was explained by Alfred Stewart, as I had already explained to you. Okay, so NEET 2016 phase 1. In a test cross involving F1 hybrids, my dear students, more parental type of an offspring were produced rather than the recombinant type. So what is this? This is what, my dear students, Morgan was talking about in his experiment over Drosophila, where he discovered linkage and recombination, and he said, my dear students, whenever you're taking two genes into consideration, and if they are present on the same chromosome and they are present at a very less distance to each other, my dear students, they tend to inherit togetherly. So togetherly. Assortment is not so easily seen in them, right? They tend to inherit togetherly. They tend to assort togetherly. So whenever there are more parental offspring or combination, there are more parental offspring than the recombinant one, it gives a clear idea that the genes are actually quite tightly linked and present on the same chromosome. So it depends upon, if there are very much, like if there are 98% parental combination and just 1 or 2% recombination, that means the two genes are tightly linked and present on the same chromosome. And it depends upon, so it depends upon the percentage of parental combination, the percentage of recombinant, it depends upon that. So if parental is very, very high and the recombinant is very, very low, that means the two genes are very tightly linked. And if this ratio is going to change, this percentage is going to change, that means the linkage is going to become weak, right? Yeah. So whenever there are more parental cross than the recombinant one, that means the two genes are linked and present on the same chromosome. Next question. Next question is the term linkage. In fact, many times, my dear students, NTA has used this question in identify the correct match or identify the correct column, like that, right? So multiple times this question has been used. So the term linkage was coined by Morgan. Next question. This was asked in NEET 2013. Which of the following statement is not true for two genes that show 50% recombination frequency? Now see this, 50% recombination frequency, that means 50% of the offspring are recombinant. 50% are recombinant. What does that mean? Which of the following is not true or wrong? So not true or wrong is the genes cannot be tightly linked. If the genes are tightly linked, they cannot show 50% recombination. It will be very, very less than that. The genes cannot be tightly linked. The genes can show independent assortment, yes. If the genes are present on the same chromosome, they can undergo crossovers in every meiosis, and the genes may be on different chromosomes. So many possibilities are possible, but the point is this is not possible. Next question. This was asked, my dear students. The ask here is not mentioned, but this is an amazing question, and this is in one of the recent, maybe if I'm not guessing it wrong, it will be somewhere around 2019 or maybe 2020, right? I think it was in 2020. Which of the following characteristics represent the inheritance of blood group in humans, right? So yes, this is the same question, uncle, that you were talking about. So the ABO blood group is an example of multiple things. So ABO blood group can also show dominance. So how it will show dominance, my dear students? Whenever it is the case of IA, IB, whenever it is the case of capital IA and small I, capital IB and small I, both these represent dominance, because capital IA is completely dominant over small I and capital IB is also completely dominant over small I. That is why the blood group over here is A and here B. Codominance. So human blood group system also give you an example of codominance when the genotype is IA-IB. So here AB blood group is expressed, both the alleles are equally dominant over each other in the heterozygous condition. Yes, it is an example of multiple allelism also, because there are more than 2 alleles. 3 alleles are possible. Incomplete dominance, no. Polygenic inheritance, no. So what is the right answer? 1, 2 and 3 are the one that represent the characteristics of human blood group system. Is this clear, uncle? Savita, my lovely kids, is this much clear? This was asked in NEET 2016. Again, very amazing question. Is this much clear, everybody in the class? My lovely kids, is this much clear? Okay, so dominance means, my dear students, whenever in a heterozygous condition one of the allele express itself. So dominance means in a heterozygous condition organism only one allele is expressed. Like in capital T small t, only capital T expresses and the organism is tall. Pea plant. Codominance means in a heterozygous condition both are expressed. In a heterozygous condition both the alleles are expressed. Both allele expresses. Next is pleiotropy. Pleiotropy means a single gene is going to affect multiple phenotypes, multiple characters. So a single gene influences many characters is pleiotropy. Polygenic inheritance, multiple genes will control or try to present a single character, right? So many genes influencing a single character is polygenic inheritance, just like your human skin color, human height, IQ, right? So what can be the right answer? A goes with 2, A goes with 2, B goes with 3, B goes with 3, C goes with 4 and D goes with 1. So D is the answer. Excellent, everybody. Excellent, uncle and Savita. Next question. Next question was in 2015, a pleiotropic gene. Okay, so a pleiotropic gene, I have already explained you, a pleiotropic gene controls multiple traits in a individual. Example of pleiotropy is, my dear students, phenylketonuria. Other example of pleiotropic gene is also, my dear students, the gene B that is present in a pea plant. That single gene B can control the size of the starch grain as well as the shape of the pea seed. So a pleiotropic gene is the one that controls multiple traits in an individual. Clear? Next question. This was asked in 2019. Select the incorrect statement. Now these are the questions that will be related to sex determination, which is also one of the important topic. Any guesses over that? Everybody just give it a try. Select the incorrect statement. Select the incorrect statement. Incorrect statement, yeah. So let me help you out then. Male have their sex chromosome much shorter. Yeah, so one of the sex chromosome is shorter because male have X and Y is shorter than X. That's absolutely correct. Male fruit flies heterogametic. Yes, that's absolutely right, just like human males. In male grasshopper, 50% of the sperms have no sex chromosome, yes, because it is the condition of XO. And in domesticated fowl, that means in birds that you domesticate, right? Like hens, sex of progeny depends on the type of sperm rather than egg. No, this is wrong. It depends upon type of egg rather than sperm, because in birds, my dear students, female heterogamety is seen. Female heterogamety heterogamety is seen. So what do I mean by that, my lovely kids? So I mean the female is going to be ZW and male is going to be ZZ. So that means sperm will be of one type and egg will be of two type, right? So two types of eggs, hence sex depends upon the egg and not upon the sperm. Clear? Next question. So this one is incorrect. Next question. This was asked in NEET 2018. Which of the following pair is wrongly matched? Which of the following is wrongly matched? Starch synthesis in P is an example of multiple allele. No, this is in fact wrongly. Starch synthesis in P can be an example of pleiotropy, that gene that is doing, or it can also be a case of incomplete dominance. So whenever this capital B, small b, my dear students, what it will produce an intermediate. I'm talking about a gene B which is present in a pea plant, which is responsible for giving size to the starch grain in a pea plant, right, in a seed. So it will produce intermediate. I missed it. So it will produce intermediate size of starch grain, right? When it is capital B, starch grain is big. Small b, starch grain is small. Capital B, small b, starch grain is of intermediate size. But if I consider the same gene is responsible for controlling the shape of the seed also, but capital B, small b in the heterozygous condition will be producing round seeds only. Right, so it is an example of incomplete dominance whenever you take into consideration size of the starch grain as a phenotype. When you consider the phenotype, shape of the seed, it becomes a dominant gene, right? So it is an example of pleiotropy, and it can also show incomplete dominance in this case whenever size of the starch grain is taken into consideration, but it is not the example of multiple allelism. ABO blood group codominance, right? Exotype of sex determinants, grasshopper, right? Now we have seen that, right? And TH Morgan linkage, that is absolutely correct. I told you multiple time NT is asking question about TH Morgan linkage, and even in your chapter number 6 molecular basis of inheritance, there are also multiple questions where NT has mixed this information of Morgan linkage into their select the incorrect match or pairing, like that questions. That's important. Next question. NEET 2020, select the correct match. Kids, you need to select the correct match. So can you try this one? Select the correct match. Can you try this one? Select the correct match. Hemophilia, Y-linked. No, my kids. Hemophilia is definitely sex-linked, but it is X-linked. X-linked recessive genetic disorder. So this is incorrect. Phenylketonuria is autosomal recessive. Sickle cell anemia is autosomal recessive trait and is linked with chromosome number 11 genes. That's right. Thalassemia, X-linked. No, thalassemia is also autosomal recessive trait. Right? So this is the correct one. Thalassemia is also autosomal recessive. Right, kids? Okay. Next question. So I hope I provided you the answer. The correct match is sickle cell anemia, autosomal recessive trait. Next question. What is the genetic disorder in which an individual has an overall masculine development but will have gynecomastia and will be sterile? Any guesses over that? Everybody in the class, any guesses over this? Come on, it's the condition of Klinefelter syndrome. Yes, it's a condition of Klinefelter syndrome. Instead of 46 chromosome, the male will have 47 because there will be one extra copy of X chromosome. Extra copy of X chromosome, because of which gynecomastia, sterility is coming. Next question. 2018. A woman that has an X-linked condition on one of her X chromosome, this chromosome can be inherited. So they are talking about a female, and she will have definitely 2X chromosome, and the genetic disorder is linked with one of the chromosome. Let's say it is C. Let's say it is color blindness, right? So this is the condition. So this chromosome, this diseased chromosome, the chromosome that is having defective gene, this is the chromosome having defective gene, can be inherited by. So let me tell you, my dear students, if I talk about a male progeny or if I talk about a female, if I talk about a male progeny or if I talk about a female progeny, female needs 2X and male need 1x, right? So in a female, 1x comes from father, 1x comes from the mother. So the mother can give this defective, yes, female can also get it, daughter can also get it. And if I talk about the son, my dear students, X, the son will get 1x chromosome definitely from the mother and the other Y from the father. So this mother can give either of the X chromosome to the son also. So both the son and daughter can actually receive or inherit the defective X gene. Both can do so. Clear, guys? Is this question clear to everybody in the class? Everybody, is this question clear? Let's take the next question then. NEET 2017. Thalassemia and sickle cell anemia are caused due to a problem in a hemoglobin molecule synthesis. Select the correct statement. Let me tell you what was the concept over which this question was asked. Thalassemia, my lovely kids, is a quantitative character or quantitative disease that is linked with the quantitative character of the hemoglobin, defect in the hemoglobin, right? So let me tell you, sickle cell anemia is a qualitative. How? Qualitative genetic disorder. How? So listen, my dear students, thalassemia is because hemoglobin molecule are produced in lesser amount. Sickle cell anemia is because the hemoglobin is produced in enough amount, but it is not correct. It is incorrectly formed hemoglobin molecule, my dear students, because in the beta globin chain of the hemoglobin, my dear students, on the 6th position instead of glutamic acid there is valine, because GAG got replaced by GUG. That was the problem, right? And because of which the hemoglobin molecule, beta globin molecule become defective, making the hemoglobin molecule defective. So that's a qualitative error. So sickle cell anemia is a qualitative genetic disorder and thalassemia is quantitative, because here the number of globin molecules are less and here the quality is compromised. So if I talk about which one is correct, yeah, second one is correct. Thalassemia is due to less synthesis of globin molecules. Thalassemia is due to less synthesis of globin molecule. Yes, is this question correct, uncle? Is this question understandable to you, uncle and Savita? Is this okay, my kids? Is this okay? Is this okay? Please tell me in the chat box. Okay, next question. This was asked in NEET 2017. A disease caused by an autosomal primary non-disjunction. So actually, my dear student, it is the Down syndrome. Why it is the Down syndrome? Because Down syndrome is an example of aneuploidy. Down syndrome is an example of trisomic condition. It's a trisomy of chromosome number 21. Chromosome number 21. So there is an additional copy of chromosome number 21 that is present in this particular type of a human. So the number of chromosome, the total, instead of 46, they become 47. It's a trisomic condition. That's because of the non-disjunction of the chromosome. Clear? Clear, guys? Clear? Can we move to the next question? I think we have come to the end of the session, if I'm not wrong. Okay, maybe, no, maybe 2 more questions. Okay, 3 more questions we have. Okay, fine. So let's take this one. NEET 2016 phase 1. Put the correct statement. Hemophilia is a sex-linked recessive disease. Do you think so? Yes, it is in fact X-linked recessive disease. This is not wrong, this is correct. This is correct, but I'm showing it is X-linked. X chromosome linked. Defective gene is present on X chromosome. Down syndrome is due to aneuploidy. It's a trisomic condition of 21st chromosome, the additional copy. Phenylketonuria, this is also correct. Phenylketonuria is an autosomal recessive genetic disorder. That is also right. Sickle cell anemia is X-linked. No, sickle cell anemia is also autosomal. It is also autosomal. This is wrong. So how many of them are correct? 1, 2 and 3 are correct. Right, kids? 1, 2 and 3 are correct. Right, my lovely kids? Next question. 1, 2 and 3 are correct. 4 is incorrect. Any doubts here? Any doubts here? Uncle, Savita, is this question clear, my lovely kids? Is this question clear, my lovely kids? Is this question clear, my lovely kids? Okay. So 1, 2 and 3 are correct. Fourth is incorrect. Next question. Which of the following most appropriately describes hemophilia? So yeah, it is a genetic disorder and it is a X-linked gene disorder. It's an X-linked gene disorder. That means the defective gene is present on the X chromosome. Okay, so uncle, you're saying no for any doubt you don't have or you did not understand. Please clarify, my dear. Next question. A human female with Turner syndrome. This was asked in 2014. A human female with Turner syndrome. So a human female with Turner syndrome will have actually one less X chromosome. So a normal one has 2 X chromosomes. She'll have one less X chromosome. So instead of 46 total, she'll have 45. So this is a condition which a female has 45 chromosome with XO condition, one less. O represents absence of one X chromosome. Clear, kids? Clear this? Next question. So I think we have now finally come to the end, my dear students. So that's all I wanted to discuss. These were the important questions that NTA has asked in the recent history of NEET exam with respect to this chapter. That's a very important chapter, and definitely 3 to 4 questions minimum you're going to get this from this particular chapter. So keep on learning, keep on revising, and do let me know if you have any single doubt. Maybe in the next session also you can let me know, my lovely kid, if you do not have right now, right? So keep on solving more and more questions, and do not forget to give a nice reading to your NCERT text with respect to this chapter. So I would like to say, my dear students, we are offering NEET 2022 Rank Assurance Ultimate Package on our platform. Do visit to infinitylearn.com website to learn more about it. And here what are you going to get is live classes, self-learn videos, self-learn flashcard, adaptive practice, rank assurance program daily test series, and also the rank assurance program test series test and doubts app. So till now, if you haven't joined our Telegram channel, do consider joining our Infinity Learn NEET Telegram channel, my dear students. Whatever we teach, whatever PDF we are using, that I'm creating, I'm writing, I'm scribbling, everything will be provided to you in that particular Telegram channel. So do consider. Thank you very much. Do consider joining it, and the link is in the description. So that's all. If you haven't subscribed till now, please consider subscribing. Give me a thumbs up, share with your friends so that others can also get benefited, and also leave a comment, my dear students. Do let us know, how do you find this session? Do you find this session helpful? Do let us know in the comment section. Thank you everybody for joining the class. Thank you Savita and Uncle. Thank you for participating. That's all from this session.